Alright, back at it again with another exercise application assignment. I'm your host, Graylin Moon. And let's get into it. First, let's cover training. Today was lats and triceps focused, so that means had a frontal plane pull, which was my weighted pull-ups, wide grip, kind of starting the bar action. Of course, I think I mentioned before, when you are adding weight to it, it kind of makes it hard to go real strict sternum to bar, but then, I do as many as I can with weight, then I drop the belt and burn out my body weight. Then moving on, we did I say we, it's me, I did a bilateral lat pull down. So, it's just the machine that has two cables and allows me to get my hands, kind of close and at a little bit of at an angle. Did that, nice and heavy on that, and then we did a chest supported cable row, single arm. So that's kind of chest support, good stretch, good contraction. So that covers frontal plane, sagittal plane, pulling, and as well as what would be the word? Like a pull down and a row. You know what I mean? So that kind of covers it. That's generally my approach to training lats. I like to throw in pullovers every once in a while, but that's not purely a lat exercise. It's really more like a weighted stretch. You're getting a lot of chest and you're getting a lot of serratus and other stuff. So, I'm trying something slightly new. We hit triceps as well. So that's going to be our skull crusher. Finished off with, what is it? Forced eccentrics. So I'm right here, I'm laying down on the bench, doing skull crushers, and then I can't get any more, so I bring it back here, I press it up, lower it slowly, bring it back here, press it up, lower it slowly, and then single arm tricep push downs, and then neck flexions, get that bull neck. Do that. I just do that pressing my palms into my head, and that way it's a perfect resistance curve. Actually, I can force it down harder on the eccentric and then apply less force on the concentric, so that I'm really working the crap out of my neck. I wish all exercises were like that, where it's very, very hard lowering the weight, and then it's becomes easier and easier to, or like as you're lowering the weight, it gets heavier and heavier and heavier, and then you push up and it gets heavier and heavier and heavier as you push up. Really? Okay. You want your strongest contraction out of the bottom of the hole of like a squat or something like that. But you're weaker on the contractile portion of the movement anyways, except for like at the very top of the range of motion, because everybody can quarter squat a million plates. So it would make sense that it gets harder there as well. But, I'm getting off track. I'm just saying that it would be nice if everything could be kind of self-regulated in a sense. But let's talk about lecture. So today we kind of covered a little bit of chapter 8, talking a bit more about the sliding filament model. Really the notes that I took covered more of the structure of the muscle. I didn't quite make it all the way through chapter 8. I know I should have, but I had other work to do. So is what it is. And also I kind of puttered about a bit this weekend. So, I drew 13 gorillas because it was really fun. I drew them with crayons, and then I did some other drawings and played Uno with some guys. So, I need to get on it. But I made it about halfway through the chapter. Really the big thing to cover is excitation, contraction coupling, and sliding filament model, and just make sure I got that down. But I got it down enough to talk to you today about it. So basically, what I'm doing in exercise today, let's think about the tricep, because it's a simpler muscle than the lat. Yeah, let's think about the tricep right quick. Not the best lighting, but so basically, when I'm contracting the tricep, first of all, let's go over the types of contractions. We have a concentric, an eccentric, and a passive. So the difference is a concentric contraction means the muscle is shortening. So in my tricep, that's shortening. This is lengthening. But in the absence of load, that said, it's passive. So let's talk about my bicep. So if I'm doing my skull crusher, right? This would be the concentric, this would be the eccentric, because I'm holding a dumbbell in this hand. It's under load. What about my bicep? My bicep is certainly shortening and lengthening during a skull crusher, but it's not under load, so that's called a passive contraction. It's just kind of moving. It's just kind of being reciprocally inhibited. It's reciprocal inhibition in a sense, like it would be with a reflex. As this lengthens, this shortens, and as this shortens, this lengthens. But since there's no load associated with it, it's just passive. And it just also needs to mention that when you're eccentrically contracting, right? I'm lowering the dumbbell right here. It's still, your myosin heads are still trying to climb. They're still trying to do this action. Really, what they're doing is they're decelerating the weight. That's why you control your eccentric. That's why you're also stronger on the eccentric. It's because you're performing the same action as you would be when you're contracting in the concentric, except that you're kind of just, I like to imagine it like you're kind of just spinning your wheels a little bit. So like they're climbing, but they're not really climbing. They're lengthening, but they're trying to climb. Maybe it's like lengthen 2, climb 1, lengthen 2, climb 1. A sort of a 2 steps backward, 1 step forward type deal. Not sure the specifics, but essentially it's like hitting the brake pedal a little bit on a car. The gravity wants to make the weight go, accelerate it, 9.8 meters per second. It wants to make that weight do that, but you're slowing it down, kind of applying the brakes a little bit. So that's an eccentric contraction. Concentric simpler, you just have your sliding filament model. So what that looks like is, first of all, you have your muscle encased in a fascia, which is encased by an epimysium, which contains fascicles. Fascicles are bundles of muscle fibers. Well, fascicles are surrounded by a perimysium, which contains, bundles of muscle fibers. What do bundle of muscle fibers have? They're wrapped in an endomysium and contain your myofibrils. Myofibrils are made up of myofilaments. You have your thick filament, myosin, and your thin filament, actin, which also has this thing called, another protein on it called troponin. And when there's also these things called transverse tubules, which kind of like, if this is like a cross section of a muscle cell, transverse tubule is a hole that is continuous with the sarcolemma, and the sarcolemma can be charged. So when an action potential is sent from the neuromuscular junction, right, you have your nerve right here, the infoot, and it gets, an action potential sent through it, shoots acetylcholine into the synaptic cleft, binds to the ligand-gated channels on the sarcolemma. We start depolarizing the sarcolemma. That depolarization is going to propagate down those transverse tubules, and it's going to release, yes, because you have the sarcoplasmic reticulum, which kind of lines, which kind of like lines in between the the muscle fibers or whatever, and that sarcoplasmic reticulum terminates every so often in these things called terminal cisternae, 2 terminal cisternae and a T-tubule in the middle. That creates a triad. Now, when the action potential propagates down that T-tubule, what is it called? It activates a receptor, this voltage-gated receptor called dihydrogenase pyritase or something like that. I don't know. I can look at my notes, but I'm trying not to look at my notes for this. It's like this big long word. It's like dihydrogen pyramid, something, whatever. Basically, that's a voltage-gated thing, and it causes within the terminal cisternae, they start to release calcium from because this other receptor gets activated because of that receptor. It's like Ryan something. It's Ryan receptor. Ryan receptor gets activated. I need to really review the names, the actual names, but Ryan receptor gets activated. Then we start pumping out the calcium. And when calcium hits, That's your key. Key's gonna unlock troponin. Troponin's that thing on the actin filament, right? The thin filament activates that. Tropomyosin, changes the shape, right? We're turning the key lock, right? That turns the key lock, and what that happens is, then you have the myosin head, right? Of the the thick filament. This is your myosin head, and then this is your thin filament. And we've turned the keyhole, so now this dude can bind with ADP, and essentially go through a process. I need to review the specifics of the process. That can be next episode, but it binds, and then eventually it does like a little power stroke, and pulls this way, and then ATP comes along and disconnects it, right? And then ADP is right there, right? That's what happens. Yeah, that's your excitation contraction coupling. So that's also our connection between bioenergetics, neural function, and muscles, right? So you need ATP for, what do you need? Where am I going with this? You need ATP for the sodium potassium pump in order to set resting membrane potential. That way there can be an actual action potential, an EPSP or an IPSP that is sent to a muscle. EPSP in the case of you want to move the muscle, or there's a reflexive contraction. You can't see what I'm doing about reflexive contraction. Or there is an IPSP in the form of, what is it, the Golgi tendon organ, your GTO. Too much tension on the muscle. Oh my gosh, I'm squatting 1,000,000 pounds. Can't do it. Drop weight. Your muscle spindle governs the EPSP of the reflex. Golgi tendon is going to do the inverse stretch reflex, right? Which is dropping. When there's a big contraction, is going to inhibit that muscle. Suffice it to say, you need ATP in order to maintain that resting membrane potential, so that there's a difference between the cell and the outside. That's how we generate the electrical signal, from the ions, which are positively and negatively charged. So to get that, then you need ATP again when you're talking about releasing the myosin head. So that's how we connect bioenergetics. That's basically all we covered in lecture. When it comes to my training, that's happening all the time during my training. I'm thinking, ooh, yes, lower it slowly, contract. I'm using ATP, and I'm hopefully producing some sort of adaptive hypertrophic stimulus during that time. But yeah, that'll about do it for this episode. I will see you in the next one.